I'm going to be talking upon a topic that has become relevant today and a challenge in the days of so many generics, etc., that have come in, so many different medications that have come in, and we don't really know what we are prescribing to our patient, and we need to assess for ourselves and find out whether this drug is really worth prescribing and many other things about it. So quality is ultimately what matters. You ultimately want to give the best to your patient, but how do you know what is best? As doctors, when we are studying in medical school, we are not really taught to assess this part of it. We are taught about the drug, we are taught about its side effects, its contraindications, and all of that indications, but we're not really well equipped to understand how a drug is made, what are the processes that go into it, and how do we assess upon the authenticity of what we are prescribing. So today that has become very relevant because costs are going up. A lot of copies do come into the market, some good, some bad. It's not that I am standing here to speak against generics. It's just to share some kind of knowledge as to how we are supposed to assess or evaluate a particular product, on what basis we are supposed to evaluate. That is what I'm going to talk about. So I have a disclosure to make for this talk. Yeah. So as I said, quality matters, and it's not just with a drug, it's a quality of whatever service you are giving to the patient, whatever goes behind the scenes, that is the clinical research that has gone behind making of a drug, the quality of the product, as I mentioned, the quality of your treatment, what kind of patient education you are giving to the patient, and the quality of patient care, the adherence, the quality of the adherence, whether your patient actually listens to what you're saying, and last but not the least, ultimately what is important for us is the quality of life that the patient is getting despite his illness. So that is what matters to us most. So everywhere we want to do our best. It means in each one of these fields what we want is quality. So that is the underlying message over here. And what I'm going to talk to you first is about the quality of clinical research. All of us know that the plethora of drugs that have come out in the last 10 to 15 years in the world, especially in the field of diabetes, are as a result of cutting-edge clinical research. If clinical research happens, we get new molecules and we can serve our patients better. But clinical research has certain norms. It has to be conducted in a certain way. And these rules that have been laid down are after years of experience of what is good quality. And a clinical research product that has come out of this kind of a research can be considered to be a good molecule because the research has been of top quality and has been of the best standards. So let me go a little into the quality of clinical research and what are the parameters. Now this I have seen at very close quarters because I have been a principal investigator for several different kinds of clinical research, right from phase 1 to phase 4, and the different aspects that are there in a clinical research is what I'm going to talk about. So basically there is a lot of training that goes into it, there are human factors, then there are systems and analysis, there are integrated plans, audits. Audits are very important. Audits can come from the sponsor, audits can come from regulatory agencies like the FDA or the DCGI, and these are very important to maintain the checks and balances. And that is going to decide the quality of the clinical research. So if you look at this is in a nutshell of how clinical research actually happens. As you can see at the bottom panel here, everything starts with from the clinician's end. Everything starts with the approval. IEC is the ethics committee approval. So once the ethics committee approves a trial protocol, which has actually come from the sponsor or from any investigation agency, then the collection of data starts and the pooling of data, and all of that time as that is happening at the clinical end, what you can see in the top panel is validation, proofreading, generation of queries, satisfying of those queries. All of this has to go on in tandem simultaneously. And at the end of it, you get a trial report which is approved and all the analysis has happened. So this is how a clinical research trial is conducted, if it is conducted in the right way. Now you can see that at every point in a clinical trial, there is an importance of quality. If anything goes down at any of these, then the results that you get out of such a clinical trial are questionable. So they have to be very robust systems in place for ensuring this kind of quality. So this is called a risk management model. It's not very complex. Let me tell you, I'll put it in very simple words. At every step of clinical research, there are certain risk factors that threaten to damage the quality of that trial. These are called risk factors. And managing those risk factors is this risk management model. So you need a quality of risk management model that is so robust that at every step, all the risk factors that threaten a clinical trial process are mitigated. So that is the importance of having this. Like for example, you have risk monitoring and review, risk identification, risk control and tolerance limits. So all of these at different, I'm not going to go into the details, but at different points of time, there are different factors which threaten to damage the quality of that clinical trial, and you need a risk management model of a very robust quality to mitigate all of that. So that's the important part. Let me now come to a particular study called the Verify study, which is a multinational, multi-ethnicity study versus the UKPDS, the basis of most of our treatment recommendations and decisions. We all know that. So this is basically the vildagliptin efficacy in combination with metformin for early treatment of type 2 diabetes. So this is one of the studies. The background that I gave you is to ensure that all studies should undergo those kind of vigorous scrutiny. So the objective of this study is to compare the durability of glycemic control with vilda and metformin combination versus a metformin monotherapy. It's a 5-year multi-center randomized parallel group double-blind study, and the co-primary endpoints are time to treatment failure, rate of loss in glycemic control over time, and the patients that were enrolled were either drug naive or newly diagnosed with type 2 diabetes and an A1C of 6.5 to 7.5. And it is ongoing. You should be getting the results this year. So the baseline characteristics, as you can see in this particular study, are that the A1C was 6.9 plus minus, systolic, you can see all the parameters here, I won't go into the details, and the weight was around 85 kgs, and you can see that most of these patients were obese with a plus minus BMI of around 31, and the duration of diabetes was around 3.5 months. So they were either drug naive or just newly diagnosed. The microvascular complication rate at diagnosis in this study was 8%. The GFR was around 87. So these were the baseline characteristics. So this is a study that is ongoing, and it will undergo the rigorous travails of the kind of clinical research that I talked about just now. Now, what about the quality of the product? So you have a clinical research that is very good. All the risk management factors are in place. Everything is excellent. What about the product itself? How do you assess how good a product is? For that, you have to realize that quality of a pharmaceutical oral formulation depends upon, one is the intrinsic property of the molecule. Is the molecule efficacious by itself? Okay, its inherent property, is it good enough or not? And the other thing is, how is it manufactured? What's the process? And for this, I'm going to make use of two of the parameters. One is called enantiomeric separation, and the second part is melt granulation. Now the first thing that you see, the enantiomeric separation, is about the intrinsic property of the molecule. The melt granulation is just a technology of the manufacturing process of a tablet. To tell you in brief, enantiomers are two forms of the same molecule. They're just two forms of the same molecule, and in fact, they are mirror images. Now, they are mirror images, but they are not the same. That is something you have to understand. And out of the two mirror images, usually one of them has the best activity and the other mirror image has very little activity, but it can contaminate the product. So, the hallmark of a good product is that you separate the enantiomers in such a way that the one that has the best activity is present in the molecule in the highest percentage versus the other mirror image of it. Okay, so they look the same, but they behave very differently. Like I talked about this elsewhere also, it's like Sita or Gita. So those of you who are aware of that particular movie, you know that to look at, it's Hema Malini on both sides. But the very core of these two people is so different that it's difficult to superimpose them. So it's like this. So enantiomers are things that look very, very similar on the outside, but the activity is very, very different. So this is something, this is the clinical relevance of having enantiomers. You can see here that you have the molecule on top and you have the receptor below. And you can see a subtle difference in the left and right side. These are the enantiomers. And you can see that if they have to attach to a receptor, they are quite different. So they look the same, but their activity is not the same. So advantage of having a single enantiomer formulation is there is a greater selectivity for the biological target. That means there is no mixing up of the two enantiomers, improved therapeutic indices and better pharmacokinetics. Therefore, it's a waste at best to synthesize and use one which is a mixture of the two enantiomers. You should not have a mixture as much as possible, and you should separate the enantiomers, and this is an FDA policy. The FDA urges manufacturers to identify and characterize each individual isomer of a new racemic mixture. So that is what is the important thing to understand, and this is about the quality of the product. The chirality of the receptor, again, it's the same thing, that you have receptors and enzymes that are complex structures with that center, and this, what I'm showing you here, is the exact mirror image. So one is called the S enantiomer and one is called the R enantiomer. Now let's take the example of vildagliptin, and what you find here is that the molecule contains one center in the middle, giving rise to two mirror images of S vildagliptin and R vildagliptin. But we know today that only the S enantiomer is in clinical use, and if the R one comes into it, it's actually an impurity. So you can imagine that there are so many molecules of the same, so many manufacturers of the same thing. But the subtle difference here is that the percentage of the S enantiomer is what ensures the quality and the effectivity of the drug. So that is the most important thing. So I won't go more into the details because this does sound like a pharmacology class, but these are the important things to remember. So viability of the therapeutic compound depends. Now coming to the second part of my talk, and that is the process that is called the melt granulation. So moisture sensitive therapeutic compounds may be difficult to formulate into pharmaceutically acceptable oral compositions. So if there is too much of moisture, there is poor bioavailability, poor handling and adherence, and therefore formulating immediate release vildagliptin with melt granulation is a technology that has been patented and pioneered. And this is what I meant. This is the patented technology where you have a moisture sensitive vildagliptin, and then you have a hydrophobic component to it, and then it is coated. And that is how you increase the effectivity and you increase the way it works because of this particular patented melt technology. So I have given you an idea of what impurities can be, the word to use is enantiomers. So you have one mirror image that acts well, the other mirror image that is just an impurity which should not be present, and the second part is the technology with which the medicine is made. And the next part of it, you have a good clinical trial, you have good technology, you have a good molecule, but when you are manufacturing batch to batch, you need it to be consistent. And that is the key. We've heard of several examples where, because of lack of consistency, batch to batch difference, I think we have 3-4 minutes, right? Because we are actually ahead of time. So good for me, I think. So the quality, for the quality of the product, the consistency is the key. Batch to batch variation should not be there. So that reproducibility of the drug has to be there. They have to be exactly the same. So for this, enantiomeric separation is essential to achieve the maximum specificity. And the NVS patented melt granulation process allows maximum moisture retention. But as I said, consistency has to be there from batch to batch. So next, I have talked to you about quality of clinical research, quality of the product itself, the quality of the treatment. We all talk about, we know about clinical inertia, patients not adhering to treatment, and we know that the disease is progressive. And we also know that earlier we had this kind of a methodology where we went stepwise, and the earlier and appropriate intervention may improve patients' chances of reaching a goal. So that is the most important thing. So it's time to do more for us. Again, this is the study where vildagliptin and metformin initial combination has been used. You can see the 12 weeks, the 24 weeks data, the adverse reactions. And what you can see here is that in the overall population, there's a significant A1C drop, and even in the subgroup population. Higher the baseline, higher the drop in A1C, that we know for all the drugs. Next comes quality of patient education. Now this is, to my mind at least, and in my practice, this is the most important part. The better the quality of the education, better is the adherence to therapy. And there are 2 robust studies which tell us that adherence to therapy itself in a big way brings down the A1C by 1%, whichever therapy, doesn't matter what therapy. Plain adherence to therapy brings down A1C by 1%, which means to say adherence has got the capacity of a drug. Most of the oral drugs that we have today, what is their capacity to reduce the A1C? It's about 1%. And simple adherence to whichever therapy also brings it down by 1%. So that is very important, and that depends on the quality of patient education. These are just some slides to tell you about lack of awareness actually of diet, physical activity and health education. We have ADA guidelines for patient education with respect to nutrition, education and emotional health. These are just guidelines, but they have to be indigenized to suit our whatever we do at our place. So quality of patient care again is important. Even if we have very good centers, we don't have very good A1Cs, so our quality seriously is lacking. Again, quality of adherence, I have already told you non-adherence, the outcomes are pretty bad, as you can see from this slide. And what could be the reasons? One of the key reasons of non-adherence could be hypoglycemia. So perhaps we need to move to therapies that remove this particular obstacle, and we know that gliptins in general and vildagliptin also is efficacious without causing significant hypoglycemia. There are other key reasons for non-adherence such as pill burden. We know that diabetics have to take several pills. Cost could be another huge problem for adherence. So at the end of it, quality matters. So I have touched upon in detail about clinical research and quality of the product, and a little bit about the other factors that decide how a patient outcome is going to be. And with that, ultimately I told you the most important thing is quality of life for the patient. So the predictors, you can see here the various predictors for the quality of life in type 2 diabetes. There are different scores, and the ultimate motive is that and there has been, if you apply those quality of life measures, vildagliptin metformin combination has helped quality of life if you use those criteria. So thank you very much for a patient hearing, and thanks Chairperson for giving me that extra time.